Okay, so welcome to this next video in the playlist on anti-cancer chemotherapy. In this video, what we're going to talk about is antimitotic drugs, which are drugs which prevent mitosis. Okay, so these drugs are going to prevent mitosis by preventing the assembly and deassembly of microtubules. So, firstly, let's just say that cancer is a disease that is characterized. The key characterization of cancer is that the cells over-proliferate. They divide too much. Okay? There are other characteristics of cancer. The hallmarks of cancer aren't just that you over-proliferate, but over-proliferation is one of the key hallmarks of cancer. Okay, so in order to proliferate, you have to undergo mitosis. So I'm going to start off with a revision of the cell cycle and then with a revision of mitosis. Very basically, we're not going to do this in any detail, we're just going to summarize the processes. Okay? And then what we'll see is how these antimitotic drugs are going to prevent the final process in the cell cycle. They're going to prevent mitosis, and hence they're going to stop cells from being able to divide. And therefore, if you stop cells from being able to divide, then you're going to have an antiproliferative effect. And this is going to be very helpful in the treatment of cancer. Okay, so let's discuss the cell cycle. So, the cell cycle is the process whereby a cell goes from being a single cell and divides into two cells. So it goes from being one cell to being two cells. Okay, right. So, how do you undergo this process? Well, the process by which you do this is known as the cell cycle. So you start off in a phase known as interphase. Okay, so this is called interphase. And this is the phase in this original cell's life cycle where it is not dividing. So the cell is just plodding along, doing whatever it does, okay, but it is not dividing. And whilst it's not dividing, it's said to be in the interphase of the cell cycle. The interphase is also known as I phase, or also occasionally as G0 phase. Okay, so it's really not really a full portion of the cell cycle, although it is counted as the cell cycle. Otherwise, it wouldn't actually be a cycle if you think about it. We'll come to that at the end. Okay, but it's not really a portion of the active division. Okay, so interphase is the portion before you have actually started the division process. Okay, and then something happens. Something happens to take this resting happy cell from being a resting happy cell to being a cell that is going to divide into two. Okay? And there are all sorts of different signals that can do this. Okay? So, growth factors can induce this. Okay? So, growth factors. Binding to growth factor receptors and activating all sorts of pathways such as the RAS MAP kinase pathway, okay, and the PI3 kinase AKT mTOR pathway. In addition, there's the Wnt beta-catenin pathway. All of these can activate this transition from interphase to the first phase of the active cell cycle. So growth factors binding to growth factor receptors can trigger all sorts of pathways within cells. They can trigger what's known as the MAP kinase ERK pathway, which leads to the activation of certain transcription factors such as MYC and also C-phos-CJUN heterodimers, which will change protein expression enough within the cell that it will kick the cell from being in this resting phase to being in the first phase of the active cell cycle. In addition, growth factor receptors can activate what's known as the PI3 kinase pathway, okay, also called the PI3 kinase AKT mTOR pathway in full, okay, and this again leads to the activation of a very powerful transcription factor, in this case the mTORC1, the mammalian target of rapamycin complex 1, okay. They can also lead to something known as the JAK-STAT pathway, which again can move the cell from the interphase of the cell cycle to the first phase of the active cell cycle. In addition, there are other molecules such as Wnt, which can signal through the Wnt beta-catenin pathway, and again that can lead to the activation of the cell, where you move from the interphase to this first phase of the active cell cycle. So there are all sorts of pathways which can activate a cell to move from the interphase to what's known as G1 phase. Okay, so this is called G1 phase. Also, in full, this is called the first growth phase. So this is the first growth phase, also called the first gap phase. So first growth or the first gap phase. Okay, so what happens in the G1 phase? Well, basically you're going to prepare for dividing into two cells. Okay, now what do you need to do in order to divide from being one cell into being two cells? Well, firstly, you're going to have to copy the genome. So remember, in the nucleus of our cell, we have 46 chromosomes, okay? Every single one of those chromosomes is going to have to be replicated, okay? Because we're going to need 2 copies of each of those chromosomes, because 1 copy for this cell, 1 copy for this cell. So we're going to need to replicate our genome, okay? The other thing is, we're going to go from being one cell to being 2 cells, and all of these, well, both of these cells are going to need copies of all of the essential proteins. So all of the enzymes involved in the respiratory pathways, for instance, are key examples. All of the receptors in the cell membrane, all of these proteins are going to have to be duplicated. Presently, we have enough protein for one cell, okay, but we're going to need to have enough of these enzymes and receptors and whatever other proteins you can come up with for 2 cells. Okay, so we're also going to have to duplicate the proteome of the cell, which is a fancy word to mean all the proteins in a cell. Okay, so the proteome. Right, so what's going to happen in G1 phase? Well, you're not going to start replicating the genome yet. That's going to happen in the next phase of the cell cycle. Instead, what you're going to do is you're going to start producing loads of new proteins. Okay, so you're going to start with this duplication of the proteome that you need to undertake. So you're going to start making more copies of proteins that you already have, so that you've got enough protein for 2 cells when you finally do split into 2. Okay, and this is why it's called the first growth phase, because the cell is going to grow somewhat. Okay, because it's getting ready to divide into 2. Okay. In addition, you're about to start the process of copying the genome. And in order to copy the genome, you have to produce a bunch of enzymes and other regulatory proteins which are involved in that process of copying the DNA. Okay, so these proteins won't normally be expressed, otherwise you would be copying the genome all the time. So you're going to have to make those. Those are going to be made in the G1 phase. Okay, so you're preparing to copy the genome and you're starting to duplicate the proteome in the first growth phase. So, and let's have G1 phase in orange here. Okay, now what's the next phase of the cell cycle? The next phase, and I'm just going to draw this here, this is what we've got after the first G growth phase, okay? And I'll just put 1 chromosome here. Now, I can't draw 46 chromosomes out, basically, so I'm just going to put 1 there, and that represents all of the 46, basically. So, when we double the number of chromosomes, because we've copied the genome, I will draw an extra one alongside it to represent that we have double the amount of DNA as we should have. Okay, so we're now going to go through the next phase of the cell cycle, and this next phase is what's called the S phase. Okay, now the S phase stands for the synthesis phase. So S stands for synthesis. Okay, and what's going to happen in the synthesis phase is we're going to copy all of the DNA within the nucleus. We're going to make a copy of every single one of those 46 chromosomes. So let me now colour it in blue here. Okay, right. So, what happens is you begin the DNA replication and you'll end up now with twice as much DNA because you've copied every chromosome. Okay, now the identical chromosomes, so you have 46 chromosomes, each one of them has now been copied, so it will have an identical partner. They remain joined together, they don't split apart. Okay, so you get a structure that looks like this. You have 1 chromosome here, and its identical copy here. Okay, so this is the copy that you've just made. I want to stress that when you draw something like this, you are not drawing the two homologous chromosomes bound together. Okay, so let me just explain something. You will remember that we have 46 chromosomes within your nucleus. However, really it's 23 pairs. So there are two chromosome 1s. So here is one chromosome 1, here is another chromosome 1. There are then two chromosome 2s, then it goes all the way down to chromosome 22, where you have two chromosome 22s, and then with the 23rd pair it gets a little bit more complicated. If you're female, you have two X chromosomes. If you're male, you have an X chromosome and then a Y chromosome. So I'm male, so I'll draw an X and a Y here. Okay, right. So these two chromosomes joined together, they are not both chromosome 1s joined together. No, what you have done is you've copied both of the chromosome 1s. So you've got identical copies of both chromosome 1s and they're latched onto each other basically. So you've gone from what we drew originally to having this basically. You've now got copies of every single chromosome that are latched together like so. Okay. And these structures where you have two identical chromosomes latched together, they are called sister chromatids. Okay? Right. So, I'll stress that one more time. They are not the two homologous chromosomes linked together. Absolutely not. They are the two sister chromatids linked together. Okay? And the center bit where they're linked together is known as the centromere. Right. Okay. So after the synthesis phase, where we have copied all of the 46 chromosomes and now have 46 sister chromatids, or if you like, 92 chromosomes, what's going to happen is the next phase of the cell cycle, and the next phase is what's known as G2 phase. Okay, and in G2 phase, you're going to continue the noble work of G1 phase. Okay, you're going to continue replicating the proteome. Okay, so you're going to make more proteins, the cell will grow a bit more, and you're also going to produce the proteins that are going to be needed in order to go through the next stages of the cell cycle. Okay, so in the next stages, you're going to undergo mitosis. Okay, now, in mitosis, what's going to happen is firstly you're going to divide the nuclei. Okay, so you're going to go from having one cell with one nucleus with 92 chromosomes in to having one cell with two nuclei, each with 46 chromosomes in. So the next phase of the cell cycle is what's known as mitosis. Okay, here. And mitosis is, as I say, it's nuclear division. Strictly speaking, mitosis does not mean the division of the cells. It means the division of the nuclei. Okay, so in mitosis, what's going to happen is we're going to go from having one cell with two sister chromatids joined together like this for each of the 46 chromosomes, to having one cell with two nuclei, and each of the nuclei has one copy of each of the 46 chromosomes. Right, so let's colour mitosis in turquoise. And then the final phase that you're going to go through is a phase known as cytokinesis. And this is when the cell actually does split into two. So it goes from being one cell with two nuclei in to being two separate cells. Okay, so cytokinesis is what the fancy word for cell division is. Okay, right. So I'll colour then cytokinesis in yellow. Now, mitosis and cytokinesis are often grouped together and called M phase of the cell cycle. So these two together are called the M phase for the mitosis phase. But as I say, mitosis, strictly speaking, means the division of the nuclei. Okay, so, as I was saying, G2 phase, you need to produce all the proteins that are going to be involved in splitting firstly the nucleus, which has these 92 chromosomes in, into two separate nuclei, each with 46 chromosomes in. And then you also need to produce the proteins that are going to be involved in splitting one cell with two nuclei in into two separate cells, okay? So you're going to produce the proteins ready for mitosis and cytokinesis. Okay, so that's the basic picture of the cell cycle now. what we're going to do is now zoom in on M phase, okay? So we're going to look at mitosis and cytokinesis in more detail, because these antimitotic drugs, they're not going to target all of this bit. They're going to target this bit here. They're going to target mitosis specifically. Okay, right. So, mitosis has many different stages in it. Okay, so let's start with our picture of a cell that has just come out of the G2 phase. So it's a nice big cell. It's got a nucleus here, and this nucleus has 46 sister chromatids in. So 46 of these structures where you have two identical chromosomes joined together, and they're now called chromatids, but we'll still refer to them as chromosomes. People know what you mean if you use either chromosomes, chromatids. Okay, so we have 46 of these. So let's draw another one here. I'm going to have to draw a few of them. I'll draw 3 of them, but in actual fact you'll have 46 of them. I'm not going to draw all 46 out, but I'm going to draw at least more than one, because it will look pathetic if I don't draw more than one. Okay, so this is our cell that's just come out of G2 phase. Now, the first thing that's going to happen is prophase, okay? And what will happen in prophase is 2 things that aren't going to look very motivated from the picture that I've drawn. Okay, so in prophase, which is the first phase of mitosis, 2 things are going to happen. Firstly, you're going to get chromosomal condensation. Okay, now this looks totally unmotivated from what I've drawn. Okay, so I have been drawing all along these condensed chromosomes already. Now the reality is they were not condensed, okay? So through all of this phase here, the chromosomes never ever had these structures that I've drawn here. Instead, what they looked like was this, okay? So in the nucleus, instead everything looked like this. You have just DNA everywhere in no sort of order whatsoever, and this is what's known as chromatin. Okay, now for obvious reasons I did not draw chromatin here, because it would have been useless as far as understanding is concerned. I drew the condensed chromosomes because the human mind finds it a lot easier to understand those. However, this exact same thing we've been discussing here, where you had replication of all of the chromosomes to produce these identical copies of one another, this was happening, it was happening with these squiggles rather than with these nicely condensed structures. Okay, now in prophase, what is actually going to happen is the chromatin is finally going to condense, and you will finally get these structures forming. So you will finally be able to see 46 sister chromatids together. So you're not actually going to see these sister chromatids until you finally do get into prophase of mitosis. Okay, so before that it just looks like a squiggly mess like this. Okay, so in prophase the chromosomes are going to condense, and then you'll finally see these 46 pairs of sister chromatids joined together. Okay, then what's also going to happen is you're going to start breaking down what are known as the microtubules. Okay, right, and again, this is something that seems very unmotivated from the picture that I have drawn, because I haven't drawn any microtubules. What am I on about even? Right. So, in cells, you have to ask yourself, what is actually holding a cell in the shape that it is in? Why do cells stay in the same shape? Well, it's not the cell membrane. Even though we draw pictures of cells, so I'll draw a picture over here. We draw pictures of cells like this, with a cell membrane and then a clear cytoplasm here and a nucleus here. Okay? Even though we draw pictures like that, in reality, this is very, very false. We do it because it helps our minds understand it. But the reality is, what is holding this cell membrane up? This cell membrane is goo. It's not got a shape. It will just mold into whatever shape it wants. So why do cells maintain a constant structure? Well, it's because inside them, you have a meshwork of protein filaments, basically, everywhere. Okay, so you have protein filaments everywhere. Okay, and again, we don't usually draw this because this is what it would look like if we actually drew it. Okay, a squiggly mess. Okay, so it's not very helpful, but you should remember that there is this protein meshwork everywhere within the cytoplasm, which is holding the cell membrane up. Now, this protein meshwork that is within the cytoplasm is what's known as the cytoskeleton. Okay, now the structures that make up this cytoskeleton are what are known as protein filaments. Now, there are two major types of protein filaments that make up the cytoskeleton, okay? And these are actin filaments and tubulin filaments, okay? So actin is one type of filament, and tubulin are the other types of filaments, okay? And actin, basically, and tubulin, they're both tiny little proteins, but they polymerize together to make huge, great, long fibers, okay? And these structures, these actin filaments and tubulin filaments, and by the way, tubulin filaments are also known as microtubules. Okay, these make up part of the cytoskeleton of the cell, basically. Now, microtubules are a normal component of the cytoskeleton. Okay, so when I say we're going to break down the microtubules, that's what I mean. The normal microtubules that are within the cytoskeleton are going to be dissembled. They're going to be broken down to monomers, okay, and that's going to deassemble the cytoskeleton. Okay, now, that's what happens in prophase then. You've got the chromosomal condensation, and you've also got the breakdown of microtubules, okay? And also what's going to start happening